Hey everybody, we are here for our first lab lecture, I guess, about microscopes. A lot of this will probably be review for you from Biology 101, but if you haven't taken that in a while or you just want a little more in-depth information while you're filling out your lab worksheet, then this is the lecture for you. So let's just dive on in. So our first part is, how do you take care of a microscope? You want to pretend that your microscope is like your science child. It's your firstborn and you protect it at all costs. So when you carry a microscope, you place one hand on the base, that's the bottom part, and one hand on the arm. That's the part that holds up like the oculars, the part where you look through with your eyes. You hold it close to your body by those two parts. And you walk slowly, you set it down gently, and then you're able to use it. And we do that because this equipment is a little bit delicate and can be a bit expensive. So we want to be careful with our microscopes. When we clean them, you use just a lens cleaner and lens paper on our objectives or the ocular lenses. You never ever use a washcloth, a tissue, a Kimmy wipe, a hanky, a shirt sleeve, alcohol, um, dish soap, none of that. You just want to use the lens cleaner and the lens paper. It's specially designed so that it doesn't leave any dust and that it doesn't scratch the lenses. Then, of course, before you put your microscope away for the day, you want to put it back onto the scanning objective. That's the red objective lens. You want to clean the objectives and the oculars, and you clean those objectives and oculars before you use the microscope and then before you put it away. You don't want to trust that someone cleaned it before they put it up, even though you're supposed to, and you don't want to get any sort of germies on you from it not being cleaned. Of course, you turn it off, you wrap the cord around the base, you put the dust cover back on it, and then you put it in the cabinet that it came from. So here's a picture of what we mean when we say to carry it. You see one hand is on the base, one hand is on the arm, it's close to the body, and this person is walking it to their desk so that they can use it. The most common type of microscope that we are going to be using in biology is a compound light microscope. And this is what that looks like in our labs. You can also see down on the bottom corner of our slides here that there is a microscope there that only has one ocular lens. That's a very common setup, but the ones that we have at Trident have the two ocular lenses, which are really nice to be able to look into and see your specimen. So the next thing that is most useful for you as you are filling out your lab worksheet especially is this slide and the next slide. If you are someone that does really well with picture visual learning, then this slide is probably going to work for you. I'm going to tell you about all the parts and pieces and what they do. But if you're not someone that likes that type of learning, then this second slide will be the one for you because it will have the name of the piece, where you find it, its description, and what it does. So I'm not going to go through both of these slides in their entirety because they're the same information, just two different ways. So let's go back to this picture and go through our different parts and pieces and what they do. So let's start up here at the top. These are our oculars or ocular lenses. That is what you use to look into the microscope to see your specimen. For everything that we do in this lab, we're going to say that an ocular lens has a 10-time magnification. These oculars can be changed in a microscope. You can take them out and put stronger magnification or less strong magnification ocular lenses in. So depending on what you're looking at. But again, ours will be 10 times magnification. Sort of right next to them, below them, is the ocular focus. If you are looking at a specimen under a microscope and it looks a little blurry, you can start adjusting that ocular focus. And you can also adjust how close and far apart these two oculars are. And that just allows you to see more clearly the image of your specimen. If we sort of go to the right on our microscope, you'll see this little boxy piece with a little knob. That is the rotating binocular head and the binocular head rotation knob. Basically, that is there that you spin that little knob, and then this whole set of oculars can rotate around the microscope. It's very useful if you are sharing a microscope with someone, or if you need to move around the table that you are at. You don't have to pick up and turn the microscope. You can just turn the lenses. If we keep going to the right, we see the arm. Of course, that's the piece that you want to hold the microscope by with one hand, and that is holding up this ocular lens and this rotating binocular head and all of that stuff. If we go down all the way past the arm, you get to the base. And the base is the other place that you hold a microscope when you're carrying it. It's what you set down on the table. And it's just going to hold up the rest of the microscope. In the base, you will see that there is a power switch. That's how you turn it on and off. And right next to it is a light intensity adjustment. It's like a little spin dial that will allow you to make the light be brighter or dimmer on your microscope. Okay. Sort of going back up and back to the left, you'll see this stage or platform. It's just a flat place where you put the slide of your specimen. On that stage or platform, you will see a stage clip or a slide holder or a slide clip. All of those words mean the same thing as you're reading different literature or watching different videos. All mean the same thing. And that is just where you put your slide with your specimen. It will hold it in place. Underneath that clip is going to be the mechanical stage controller. These are a set of knobs that you can turn that will allow your slide to move forward, backwards, left, and right. Some microscopes do not have that feature, and you actually have to move the slide with your own fingers, but we have microscopes that have a stage controller. Next to the stage controller, you're going to find the fine focus and the coarse focus. The coarse focus is the larger knob, and that is only used when you are on the lowest power, the scanning, the red objective lens. You only use it when you are on the lowest power. The fine focus is what you use after you've switched to a higher magnifying objective. And both of those focus knobs are for exactly what they say, to focus the image of your specimen. If we sort of go back up to right underneath our oculars, you have a very fun piece called the rotating nose piece. It's a little disc that holds the objectives. And then you've got your objectives. Those are your lenses that allow you to magnify your specimen. And we have the red one, that's our lowest, that's the scanning, where we can use our coarse focus. You've got blue and yellow, and then an oil immersion lens as well, all connected there, that are higher magnifications. And we'll talk more about those as the slides continue. Right underneath that stage, that's going to be where you find your sub-stage condenser. In that sub-stage condenser, there is going to be a little knob. That's your condenser knob. And there's going to be the iris diaphragm, which is basically an opening or a hole that allows you to adjust how much light comes in. So you can adjust the light with your light intensity knob, or with that iris diaphragm in the substage condenser. Mostly the goal of that piece is just to focus the light from the light source. And then, of course, you've got your light source. For our purposes, that's going to be a light bulb that needs electricity, but there are microscopes that use mirrors instead, that are often used if you're going like out into the field, to get your samples and want to look at them right away. Okay, again, this is all of that exact same information, just in a chart form. So if you are someone that likes charts, great. This will help you fill out your lab worksheet. If you like pictures and listening, then the other slide will work. So let's talk about magnifying power. Really, that is just how many times larger an object will look compared to its actual size. So if we went to the beach today and we looked out and we saw this image, the first one, you don't really know what's happening. You can use context clues and imagine it's someone surfing, but that is what it would look like to your unaided eye. That is 1 times magnified. If we could zoom in like a camera lens on that object, we would see that now we can make it 7 times larger. And if you wanted to, you could measure the height of this little black dot that is the surfer. And then you could bring that measurement over here and you would see that this surfer is 7 times bigger than this one, and 8 times and 10 times, and all the way till we get to our 20 times larger in this bottom picture. And you could measure, if you wanted to, the height of that first surfer with your unaided eye to how much larger he is in that last photo, and it would be 20 times. You'll also notice as you're magnifying, you lose the background image as you zoom in on the thing you're making larger. So we'll talk about that as we move forward in the slides, but that is something you can observe here. How do we figure out how much our specimen has been magnified? Well, we need a little bit of math. It's nothing too crazy, but we will take the magnification of our ocular lens, and we've already said for everything we do, that is 10 times. And then we will multiply by the magnification of the objective lens, and that will get us our total magnification. So again, let's look at those lenses. The ocular lens is 10 times magnification. The red or the scanning objective, that is going to be 4 times. Then we have our low power, which will be 10 times. And then our high power is 40 times. And the oil immersion is 100 times. So how could I find the total magnification of an object that is being viewed under low power? Well, we would take our 10 times ocular magnification, multiply by 10 times our low power magnification. 10 times times 10 times gives us 100 times magnification. And there are some practice problems for you, sort of in your lab worksheet. But if you are just wanting more practice, there's also some videos on the D2L to help you get this concept. Okay, field of view is next. Field of view is just that illuminated circle that you see when you look into the microscope. So if you've looked into a microscope before, you know that there's this area that you can see. That's your field of view. And we can see here this little water flea takes up the entire field of view in this photo. We can calculate the diameter of the field of view, or we know the diameters of different fields of views. And the diameter is just the straight line distance across from one side to the other. The higher your magnification, the smaller that diameter is. So it's sort of like using the zoom feature on your camera or your phone. If you want to zoom in on an object like a person's face, as you do that, the total area that you are viewing gets smaller. Because of this, you always want to start and always end your session with a microscope on that lowest power or the scanning objective. That way, you and the people that use the microscope after you get the largest field of view. They're able to find their specimen before they zoom in. And here's a picture sort of showing what we mean. So we've got our 4 times um, objective lens here, our scanning lens. And if we looked at something that's like a little line with checker pattern on it, under that objective, you would see the bottom picture, like black and white squares all the way across the field of view. As we move up in the second picture to a 10 times objective, you can see now that we can't see all of those black and white squares. We're zooming in. And then finally, on the 43 times magnification, we just see a black square. So as you zoom in, you have a smaller diameter to your field of view. This chart will be very useful to you as you are filling out your lab worksheet. It gives you the different objective names, the color band that they have, what their magnification is, the total magnification when they're paired with that 10 times ocular lens, and then how big is that diameter. So for example, our red is our scanning objective. It has a 4 times magnification by itself. With the ocular lens, it's a 40 times magnification. And the field of view is 4,000 micrometers. This weird little U-shapey thing is micrometers. And again, as you move up in magnification, like to the yellow, you can see that we now have 100 times magnification. We only have a 2,000 micrometer field of view. And when you get to something like the oil immersion, which has a black or a white band, you're only going to have 200 micrometers, so much smaller than our red scanning objective lens. If we know the diameter of these lenses, then we're able to figure out how big our object that we are looking at is. Now, there are slides that are specifically made that have grids that you can get very accurate counts. But for our purposes here, we're going to just use estimation. In order to estimate the size of the object, all you do is estimate about how much of that field of view is covered and then multiply it by the field of view diameter. So for example, if an object like that little water flea at the beginning of this little section stretches all the way across the field of view, then it's the same length as the diameter of the field of view. So whether that's 200 micrometers or 4,000 micrometers, whatever objective lens you're on, if the object goes all the way across, that's how big it is. If an object only goes about halfway, then it's half the diameter. So we can take 1/2 or 50%, multiply it by the diameter of the field of view, and that gives us our length or our size for our specimen. So let's look at an example. Let's say I want to know how big this blue specimen is. If I look at that blue specimen, I can say that it takes up about 1/3 of my field of view, right? So I could put 3 of them across my field of view. If I then know that I'm looking at this specimen under low power, I know that that has a diameter of 2,000 micrometers. So then what I can do is I can take 1/3 times 2,000 micrometers, And know that one of these blue oval specimen is about 667 micrometers. Next up, we've got resolution or resolving power. And that is just the ability for us to distinguish two separate points. Our eyes, they do a pretty good job. We can figure out that there are two separate points about 100 micrometers apart. That being said, our microscopes do a much better job because they can have a resolution of only zero point 0.2 micrometers apart. So that means that a regular common compound microscope has a resolving power that is about 500 times better than the human eye. Now, if you've taken any science classes before, you know that a compound microscope isn't the only kind of microscope. And that electron microscopes are used to get very high resolution and to see very small things. And this picture at the bottom sort of shows you that. This is what a radiolarian, we'll learn about this as we start talking about protists. This is what it looks like under the microscopes you would see in lab, and what your specimen pictures will look like when you draw them for your worksheets. If we could get our hands on an electron scanning microscope, which are usually only at major big universities, take up like half of a basement, they're huge, then you would see the picture on the right? Much more detail in distinguishing different parts and pieces of that organism. That's what resolution is. We also need to talk about the depth of focus or the depth of field. And that's just the distance that is in focus at any time. So you'll notice that when you are zooming in on a specific specimen, the other stuff around it will go out of focus. And that the higher your magnification, sort of the thinner or smaller that layer is that can be in focus. So you can't really start on a super high magnification because you won't really see anything because of that depth of focus. So it's another reason that you want to start on the lowest scanning objective. That way you can see a deeper and wider view of the specimen before you zoom in. And here's just some examples like in your everyday life that you might encounter, right? This little green string with these droplets, that is in our depth of focus or in our focus field. So we can see that, but the upside down face behind it is blurry. Or with a camera, we're focusing on this tennis player here. This tennis player and the background are blurry. Another thing that we need to look at is contrast. That's the difference between light and dark in an image. We adjust this with the iris diaphragm, though sometimes you'll need to also adjust it with the light intensity knob. And this picture here just shows you really the same specimen, but with different light contrast to show you that you will have to adjust this to see different parts and pieces of your specimen. So something like this far left is very bright. And if we just want to see sort of the outside membrane or this nucleus membrane, or maybe some of the things around it, then that works okay. If we get our phase contrast, then we are now trying to focus in more on just these two specimen here. We have a very defined line of light and dark where we can see the membranes and the structures. And then something like this is more dark, that you can't really distinguish the outside membranes as easily, but now we can maybe see some of the other features that are inside of these specimen. So it will really just depend on what you're looking at and what you're trying to accomplish, how you change your contrast. The great news for you as an online course right now, that's already really taken care of for you as you are looking at videos of specimen. So, not a huge thing to spend a lot of time on, but as you get the chance to practice with microscopes, you know, maybe we, can go back into lab at some point, or maybe you just have another course that has microscopes. This is something you can play with. Okay, let's look at parfocal and parcentered. Parfocal just means that all objects are adjusted to the same depth of focus. And that's really a fancy way to say that once you have focused your specimen on the scanning objective, like you've got it in view and it's clear, when you change the objective lens to get to a higher uh, magnification, you won't really need to change the focus very much. That is why you do not use the coarse focus on any objective except the lowest power, that 4 times or red objective. Then par centered, that just means when you change objective lenses, what you are looking at stays in the center. So when you get a specimen, you want to use the scanning objective to get it focused, but you also want to place that specimen right in the middle of your field of view, because when you change the objective lens, then it will not move. It will stay in the center. Quickly, a little bit about our condenser lens and the control knob. So the condenser lens is what is going to focus the beam of light from the light source up onto the specimen. And that's located right below the stage of the microscope. It has a control knob that is going to raise or lower that lens. And it's kind of on the side of the microscope or right below the stage. It moves around depending on what kind of microscope you have. But normally, that lens is all the way raised up to the stage as high as it will go. And it will help to bring that light from the light source through your specimen. Also, right there by that condenser is going to be the iris diaphragm. That is what lets a certain amount of light through, just like the iris of your eyes controls the size of your pupil. If you're looking at something that is transparent and not stained, like a bunch of pond water with little critters in it, that iris diaphragm needs to be closed down, because otherwise it will be very bright and it can really hurt your eyes and you won't see anything. It'll not have any contrast. The other way to adjust the light intensity again is to use that light intensity knob that will actually lower the output from that light bulb that is in the light source. So, how do you look at things under a microscope? Well, first, you unwrap the cord and plug it in. If it has a dust cover, you take it off. You then will make sure that it's turned on. And if you don't see a light, adjust that intensity knob. Sometimes people turn it all the way down. Make sure that you are on the scanning objective, that red objective. It should already be there, but just double check. You want to clean those lenses with that lens cleaner and lens paper. You place the slide on the stage, and then you see a whole world of things you didn't even know were there. The other type of microscope that we use in Biology 102 is called a dissecting microscope or a stereoscopic microscope. And they're pretty cool. They're not as popular in general as the compound scopes, but they're really useful. Why would you want to use this? Well, if you're trying to look through an object that is solid or opaque, you can't put light through it, then you can't use a compound scope, so you'll need to use a dissecting scope. It also will make a 3D image, which is pretty cool to see, where our compound scopes don't. They have that depth of field and depth of focus, so we don't get a 3D image. Some important parts that you want to be familiar with, you've got a powers knob. So instead of changing objective lenses to change the magnification, you spin this knob and it already does it for you inside this little tube where your lenses are at. You still have oculars that you look through. They're still 10 times magnification for our purposes. Of course, you still carry it by the arm and the base. On the base, it has a light switch, and then there are lights to help illuminate your specimen. So what are the points and purposes of Lab 1? Well, you want to be able to identify the parts and pieces of a compound microscope. You want to be able to measure the field diameters and field areas. You want to practice focusing, which really you won't be able to do for our purposes, but knowing that you use the coarse focus for the red objective and never, ever, ever again when you are on other objectives is good. Looking at some specimen, there are some videos on the D2L if you would like to look at some specimen slides. And then, of course, learning a little bit about a dissecting scope. Don't worry this before leaving. Prepare a bacterial culture. If we were in person, we would do that, but that's not the case for us today. All right. Well, that is pretty much it. If you have any questions, please let me know, and I'm happy to help you. Otherwise, thank you for watching our video, and I hope that you guys have a wonderful rest of your day. All right. Bye.